Welcome back, everybody, to the Include Data Hub webinar, Fall 2025 edition. I'm Joaquin Espinosa, leader of the Administrative and Outreach Core of the Include Data Coordinating Center. And I'll take a few minutes here to introduce the panel today. We'll be hearing right after me from team members in Include DCC who will be presenting on the latest data sets, data ingest approach, and new portal functionalities. We will then hear from Dr. Ariana Familiar about a new activity in the Include DCC related to medical image data management. At the top of the hour, we'll hear from Dr. Matthew Galbraith about a new portal, part of the ecosystem of resources developed by the Include Data Coordinating Center, the Experimental Models of Down Syndrome portal. And then we'll have a scientific presentation from Dr. James Costello on computational methods to find patterns in include-generated data sets. And we'll have plenty of time for Q&A throughout the webinar. So let me remind you that the Include Data Coordinating Center is part of the INCLUDE Project, this wonderful NIH-wide project to increase research in Down syndrome that has truly transformed the field through a strong infusion of new funding, shown here. Prior to the INCLUDE Project in 2018, the NIH investment in Down syndrome research was hovering around $20 to $30 million, and you can see that for the past several years now, we've been around $130 million. One of the key hallmarks of the INCLUDE Project is that it's an NIH-wide project with many institutes involved. These 21 institutes have funded nearly 400 projects, 14 of them being clinical trials, leading to hundreds of scientific publications. And the investment is allocated in three components: high risk, high payoff basic science approaches, cohort development activities, and clinical trials inclusive of people with Down syndrome. Within this context, the mission of the Include Data Coordinating Center is to accelerate discoveries that continuously improve and enrich the lives of all people with Down syndrome through accessible data resources that drive collaboration, connection of these communities, and the expansion of a global Down syndrome knowledge ecosystem. And one key activity, of course, is to develop, run, and maintain the Include Data Hub, which is many things. It's first and foremost a data repository, and everyone that has an Include grant is encouraged to list the Include Data Hub as the data repository of choice in their data management and sharing plans. It is a secure environment governed by NHLBI. We control access data administered through dbGaP, with the NHLBI Data Access Committee being the one that reviews the request. It's also a portal, right? You go to portal.includedcc.org, you'll experience the data hub, online, larger scale data sets, harmonized data, open, register, and control access data. But it's also, the hub is also a computing platform, and this is done in partnership with Kabatica, the platform that can provide workflows and tools, interactive web applications, and interactive analysis. If you want to learn more about data management sharing plan, there is this QR code here. If you want to learn more about Kabatica, the URL is very simple, kabatica.org. We should point out that for those interested in using Kabatica, NIH has a cloud credit program. You can learn more about it from this QR code. And there are various tiers of support, and pilot funds, limited, and then extended. And this is a great way to become acquainted with the Kabatica platform and to run analysis. And I would say that NIH is very generous on this. These are not super competitive. If you apply, you are very likely to get these pilot credits until you feel ready to start budgeting this in your grants. I would like to take a moment to highlight a couple of the resources supported by the INCLUDE project that may be relevant to this audience. One is DSConnect, the NIH-supported family registry, where families provide self-reported data through health surveys. They have access to curated educational resources. If you're an investigator and you want to promote your study through DSConnect, there's an application process that you can find here in the web page, and then your study can be promoted through these 6,000 families. And then now more recently, we have developed health snapshots and personalized care guidelines for the families. This is all funded by NICHD. And then a very exciting new initiative supported by the INCLUDE project is the SCDP Cohort Development Program, also known as Collaboration for Down Syndrome Progress. So this consists of more than 20 cohort research sites that will be employing a common protocol overseen by the Clinical Cohort Coordinating Center, DS4C, collecting biospecimens that will be deposited and shared through a INCLUDE DS biorepository, and then the data generated from this very large cohort study, probably in the thousands of participants, will be shared through the INCLUDE Data Coordinating Center. This is a large effort supported by funds from NHLBI, NIA, and NICHD. I want to put a plug here for the INCLUDE DCC office hours. If you want to learn more about the INCLUDE Data Hub, any of the tools or data sets, we host these every second Wednesday of the month at noon Eastern time. And maybe, Haley, you can put a link here to our listserv or our newsletter. If you want to be reminded of this, please sign up for our newsletter, our communications campaign, and we'll make sure that you get a reminder about these office hours. And then I would like to take a moment to acknowledge and express my gratitude for the many program officers and staff at NIH that make the INCLUDE project and the INCLUDE Data Coordinating Center a reality. Some of them are listed here, and we are thinking of them during this time of the shutdown. We hope they're all doing well. And without further ado, then I'll pass the virtual podium to my colleague, Robert Carroll, who will talk more about the nuts and bolts of the INCLUDE Data Coordinating Center and the Data Hub. Robert, all yours.
Thank you, Joaquin, and welcome, everyone. So I'm really excited to share this with you. It's things we've been talking about for a year, ways we wanted to improve the services we're offering from the INCLUDE Data Coordinating Center. This work has been a huge collaboration across all of the teams of the DCC. So I'll present and highlight a few of them, but we couldn't do it without everyone. I want to start off with, we really want to support researchers. So it's all of you, whether you're sharing data or using data about participants with Down syndrome. We really want to make sure that you have a great experience. So one of those common needs or requests that we've heard is we need citable references for the data we're sharing, right? And we all know there's a lot of benefits there. Sometimes it's purely publication requirements, right? I have to have this in the journal so I can publish my research. Other times it's just convenient to have permanent URLs pointing to those things that you can share. We want to have citation metrics and see where other people are citing your data in their work. And then again, the accessibility and reuse of data is really critical as we push fair data sharing. So with INCLUDE project data, and so that's data funded by the NIH for the INCLUDE project, that exists a lot of places right now. Sometimes it's a matter of convenience or what folks know, but it's something that we really want to change. One of the missions of the INCLUDE DCC was to have that platform where data could be centralized for the INCLUDE project and make it all available in one place. And so there's a lot of value there to bring the researchers together, to provide good tools and make this data accessible. So in order to change this status quo and help bring all the data there, that critical data, right, we need to meet the needs of investigators. And so I think this is something that we've, again, been working very hard to do over the last year, especially in a new way. And so this slide is not something new. If you've been to webinars before, you've probably seen it. And I kind of skip to the end here. Something we were doing in our first releases was really focusing on this area of metadata annotation and data standardization harmonization. And one of the things, again, that takes time. That's a lot of work, and it's really important. It creates some of those critical resources that you see when you're browsing on the portal, when you get to do that data exploration. It's very useful. But one of the things we've been doing is working on improving our release process for data sets, which have what we often term unharmonized data, but sort of source data that's being contributed, as well as minting DOIs for studies and data sets. So we want to keep, again, well, we will continue to do that important work of annotating data and standardizing and harmonizing it, but we're also we have made steps in smoothing this process out. So you're able to go directly from that data upload and sharing right out to the community. And so I'm going to cover what this looks like today, as well as a little bit about the process changes that are coming into place to make sure this all works. So the first thing that we're excited to talk about is DOIs in the Include DCC. So we have only a few of these minted so far, but we're working on expanding the count. And what you'll find that whenever you click on this DOI, which looks like DOIs you're probably very familiar with, it'll bring you right into the portal onto some public study pages. And so these are available to anyone without logging in. And you can see a lot of that same information that we saw before, details about the names of the data sets and access to those files. I guess in theory, I could just click this and it will bring us right there, right, and send me right on down to that data set. So we're really excited to have these DOIs now. One thing you can do when you're in the portal then is actually click on View and Exploration. And so what this will allow you to do is to pop right over to the data exploration tab. Now you will have to log in at this point, but it will let you see, here's all the files, right? We can see that there are 400 participants that are in these files, and we see there's about 3,200 files here. And so now if we want to get access to that data, for example, we're really interested in doing a study using this RNA-seq data from HTTP, we could check this box and select all the files, and we could analyze that in Kavatica. We could also potentially download those files as well. And so this flow then makes it very easy to say, hey, I've got this DOI, now I want to access those data. Great, now I can get access to those data files. So we're excited for that. We can also say, actually, really, I only want a single file. And so in this case, we're going to take a little bit of a deeper dive into this. Actually, we're actually going to look at the abundance file, I think, right above that. So we can click on that link and we can get information about this single file. And so this UI may look very familiar if you've done browsing before, but you can see some of the details on this page about the file, the format, the size, all that information. And you can also click to bring that into Kavatica. And so by clicking Analyzing Kavatica, it'll pop up that allows you to, once you log in, select a project in Kavatica that you want to bring those files into. It provides some helpful details to say, hey, yes, I am authorized to get this data and to bring it into the Kavatica workspace. So we've got a single file listed here, showing the studies that it's associated with. And with just a couple clicks, I can bring that into my Kavatica project. And so this is really helpful and exciting for a couple of reasons, and some of the hard work I was talking about. If I want to do analysis, now I've got this file loaded. One thing I think is key to note is, well, this didn't actually copy any files anywhere. And this is one of the cool things about the Data Repository Service, or DRS. You see that name up over here, and this is a shortcut, right? And you can see the tiny icon. This actually just said, great, now your project knows about this file that you wanted, knows the location of it. And so when you want to use it, you can access it. Kavatica knows how to access that file now. And so that's great. And so again, with a lot of these files, especially the omics files, start to get really big. You're not moving anything over. I'm not having to pay to store this file. We're just using that same copy that already existed in the cloud. One of the nice things that Kavatica has done here is they actually have started to index out of FHIR the details of this file. So now, right, we said, hey, we're bringing over 3,000 files. That's a whole bunch of stuff. Do I know where everything came from? Do I know where those files are? Where Kavatica here is bringing in the details of that, which is really handy. So this little dotted box here is just scrolled down. So there's a bunch of additional metadata that Kavatica pulls in. And so you can see all of the information about this file and the associated biospecimens have all been pulled in and made available. There's even more than this information that's there. So if you need any refresher on what the data is, where it came from, what type of analytes were used for it, you can grab all of that right here. And so I think this is super helpful. It's a great addition that Kavatica has done. And now we're in a place where we can start to analyze that file. One thing I do want to highlight, sometimes you may need, if you haven't done this before and you're bringing files in, you need to go up here in the top right, click on my name up there and add some external connections. You might need to make sure that you've connected to the Include DRS server, for example, or if you're pulling data from other platforms like Biodata Catalyst, you might need to connect that and make sure those things are connected. But that will allow you then seamlessly to bring those files into your workspace, to download them into your analysis workflows. So it's a really smooth process. Again, I don't have to pull in that data down locally and then upload it any place. I can just bring it right into Kavatica, and it handles all the complicated parts about how do I turn this DIRS URI into a place to get the data. How do I do authentication? It handles all that for you. So it's a great flow there. And so that overview again, right? You can click right on a DOI, select the files you want to analyze, bring them into Kavatica and get started. And I mentioned this before, you can bring files in from other platforms. And this is something that we're not unique in having some of these tools available. You can bring data from, again, I mentioned Biodata Catalyst before, but other platforms as well offer this exchange, offer the ability to bring DIRS URIs into Kavatica. So it's a burgeoning ecosystem there. Do make sure you link those sources that you need. If you're running into problems and you get an error, one thing that might be the problem, I ran into this last week. I forgot to relink my account. So make sure you do that to get access. So the big picture here, there's kind of a lot of complicated pieces. The DOIs, it's taken a lot of work, and there's a lot of work that you as the data submitters also will need to do to make it work. And so we've been very excited to be partnered with DataCite through a collaboration with NHLBI to give us access to these DOI minting services. And so they provide a lot of good utilities there. And we also, again, rely on you, the data submitters down here, to provide that structured metadata about the studies and data sets. And we'll talk a little bit more about that. And again, there's other partners working was a part of the data hub as well, and include DCC with Kavatica, building out the GA4GH specs so it can receive those data. Once all of these different API points and tools are in place, the flow becomes that simple one we just talked about. Go to the links and download the files you're interested in. So it's been great. It's a bunch of integration. One of the things I mentioned, DataCite is that partner. You can go to DataCite and get more information about the DOIs, right? So we mint those DOIs with them. They're the official stewards of it. But you can look and see all this information that was provided by HTTP, for example, is now available through the DataCite Commons, including that list of contributors, right? And this is something that we weren't tracking at the same level of detail before. So it's going to be important that as we continue to mint DOIs, we work with you, the data submitters, to update that information we have. There's also some stats that DataCite collects, and we're still kind of working on this. This is just a page of theirs where you can see, people are already using these DOI links here. There's a few of them who've been accessed. I think, what do we got? 45 successful, DOIs used in September. So I'm sure those numbers will go up as they're used in publications and more cited. But again, it's nice to be working with a partner like DataCite that has that tracks this information for us. I mentioned this before. We are making some updates on the ingest side. When people are submitting data, we need more information. So I realize that this can be frustrating sometimes, like, aren't we providing a lot already? But there's a lot of additional benefit here. Again, I think especially this piece about contributors, right? We have that list. I showed it before in DataCite, for example, of HTTP. Every person who contributed now can be appropriately recognized as a contributor. And I think, again, that's really, really valuable and something that we're excited to do. Also looking to capture ORCIDs if you have them. And so we can provide that information, all that metadata. So now those citations, right, your DOIs can be linked to your ORCIDs, which is great for this FAIR data ecosystem. So I hope that's motivated you to check out some new data in the Include Data Hub, to continue or consider submitting with us, to be willing and open to updating that metadata so that we could look to mint DOIs. So we're really excited whether you're looking to share data or using data to really help smooth this process up. So we'd love to hear from you if you need any help. If you run into problems accessing data, again, it's a great process, but there are some bumps, and we're constantly working to correct those problems. But if you do run into problems loading data, please do feel free to reach out, and we'll be happy to help. So again, thank you for your patience as we're continuing to build out these new services. Let us know how we're doing, what we could do to improve, and I will pass the podium over to Pierrette.
Thanks, Robert. Let me put the link. I'm putting a link in the chat to the V6 release notes that I'll be referring to. So let's see, where did my window go? Slideshow. Okay, so I'm Pierrette. I'm the data manager for the Include DCC, and I'll be going over a little bit more information about the new data from our most recent data release. So this was the V6 data release that was published in August 2025. And the link that I pasted into the chat has some very detailed release notes. So I'm not going to cover all of it today, but I just want to give a little bit of a summary and then point out a couple of ways that you can find this new data in the Data Hub. So overall, we've added 7 new study pages that give a preview for studies that have not submitted data yet. We have 4 brand new studies with both unharmonized and harmonized data. We have new data updates for 8 existing studies. We've got 6 new data types like cognitive measures, sleep data, TCR and BCR sequencing, etc. And then we have some new features in our harmonized data. So the release notes also have a section at the end where we have listed some corrections to previous data releases, some known issues that we're working on fixing, and then ways to contact the Include DCC for support. So starting with the studies section of the Data Hub, this page, which can be accessed up here under studies, gives an overview of all of the studies in the hub. And I wanted to point out that you can sort this participants column for a quick way to see which studies have data and which studies do not have any data yet. And then you can use the search facets here on the left to search for information about the studies, like the overall research topic or the general age of the study populations, for example. So I'll be talking about 2 start points to access data. One of those is through the study pages here, and then you can also access data through the data exploration tab. So the study pages are a great way to access data from individual studies. And we have a couple of major buckets of data. The first of which is what we call unharmonized data. So that is data that has not been standardized to the Include data model yet. And this could be either the source clinical data that we're sharing as it was received from our data submitters, or new data types like the cognitive measures or the sleep data that are not covered by the Include data model yet. And then we also have data dictionaries for all of those. So to access these files, you would go to the study page, and this is DS Sleep as an example. You scroll down to the data set section, and then you'll see the different data sets. And then, as Robert mentioned, if you click the Analyze in Cavatica button, these files from this data set will be sent over to your Cavatica project. And we do have documentation in the portal about how to sign up for Cavatica and link it to the Data Hub. So you can see here that there are several sleep data files available in this data set, and they each have a data dictionary associated with them. And we're also working on eventually indexing these files in the data exploration section as well. So the other bucket of data here is omics and other assay data files. And so these files are available directly in the data exploration section, where you can link them to participants or biospecimens, and there's some other file metadata that you can search on. But to access them from the study page, again, you can go to the dataset section. So this is the EXO1 Hakanarsen study page as an example. And then from the data set, you can click the View in Exploration button, or you can scroll down further to the file section where we have the files broken out by file type or experimental strategy. And if you click on any of these file counts, it'll bring up the whole list of the associated files in the Data Exploration tab. We're also working on making some additional, more detailed assay metadata available. And so this will eventually be linked here in the data set. There will be an Analyze in Cavatica button where you can push it to Cavatica and download it there, similar to the other unharmonized files. Okay, so a little bit about our new harmonized data features. So this is the participants or other data that are in the data exploration section that we have standardized to the include data model so that you can search or aggregate it across studies. So the first feature is data on interventions. So these are things like surgeries or therapeutic devices. And we've annotated the data with terms from the medical action ontology or MAXO. So they're now displayed here in the participants tab. And then over here in the search facets, you can also search for different interventions. So this is a nice addition to the diagnosis and phenotype data that we are also sharing. The next feature is measurements, and these are things like height, weight, BMI, blood labs, etc. These are not available directly in the participants tab, but for now you can download them in the clinical data download. So if you select your participants of interest and click this download clinical data button, it'll give you an Excel sheet that has several tabs, one of which is called measurements. So this is an example of measurements from the brain power study. And I've just added these colors here to highlight that we have measurements from this study for 3 different time points for each participant. So this is all the same participant, but you can see here in the age at measurement column that we've got 3 different time points. And then just to bring it back to the previous topic of unharmonized data, if you go over to the study page for brain power and scroll down to the data sets, there's one here for anthropometrics, which is those measurements, and it gives you the unharmonized source data for the measurements and a bit more information about the different time points. And then the last new feature is with our newly added DS determined study. So some of these participants were also participants in DS Connect, and it happened that we had the correct permissions to actually show this linkage in the portal. So in the data exploration tab, if you pull up the participants for DS determined, you'll see that some of them have this multi-participant icon next to their participant ID. And if you hover over it, it'll bring up this box with some information and a link, which if you click on it, it will bring up the records from the 2 different studies for that same person who participated in both. So there's one ID for DS determined and there's another ID for DS Connect. And we're working on making this more searchable in the future, but in the meantime, if you go to the study page for DS determined and scroll down to the DS Connect data set, there is a file there that you can download that has the mappings between the DS determined and DS Connect IDs for all of the overlapping participants. And this might sound familiar because we also have some studies that are linking participants using ENDAR GUIDs, which those are controlled access data that need to be accessed through dbGaP. So that's why those are not displayed directly in the portal. But if you go back to the main studies page, there's a link up here that says NDA GUIDs for Down syndrome research. And if you click on this, it'll give you a little more information about the GUIDs. And so that is it for me. Again, we have our contact info in the release notes, and please feel free to reach out if you have any questions. And now I will pass it to JP for some portal updates.
Thanks, Pierrette. Great. Yeah, so my name is JP. I'm the product owner for the portal development team. I will just open up my slides here. So I work for Dr. Freddy at the Sud Saint-Justine Research Center. Let me just open up the slides here. Great. And I'll be doing an overview essentially of the Include Data Hub, highlighting some of the new data attributes that some of the team members showed in some of the presentations here. Also highlighting some of some existing features. So a lot of the focus was regarding this new V6 data release, but I'll take the time as well to essentially explore some of the existing features, some of the new additions that we've done as well, just to help new users and also old users to essentially explore in the portal and drive some of these insights. So we'll walk through a few things here. We'll go over the landing page and essentially how we've opened up the public study entity pages. The data exploration page to build some virtual cohorts, the transcriptomic analytics focusing on a specific gene, and the variant exploration to essentially look at the variants that we have available for some of the studies that we have in the portal. So all of this focusing on a specific disease in hypothyroidism, that's going to be the focus here. So again, as a reminder, we have a feedback button on the right-hand side of all the portal pages, so you'll always see that. We definitely welcome, and more so than we encourage feedback from all users. So you could provide a screenshot, a quick message of what your feedback is. Is it an issue, etc. If you like the colors of the portal, so we really welcome everything. So don't hesitate to provide that. We also have a help center that has pretty much all the resources to explore the data hub and also the ecosystem as a whole. And you can also email support if you have something in particular. So support@includedcc.org. And as Joaquin mentioned, we have virtual office hours every second Wednesday of the month. So that's also a great resource if you have any type of one-on-one questions, and if you want to do a deeper dive with some of the data or the portal, et cetera, et cetera. So the next one coming up is November 12th. So I'll just exit here. I provided the portal website link in the chat. But you could also use the QR code if you feel like you want to use it on your phone. I think it's a little bit better on your computer though. So diving into the landing page, again, previously we've added the View All Studies page to see essentially the study repertoire of all the studies that we have. So that was really great. However, it was only in tabular format. We didn't have more information in terms of the study metadata. So now that we've unlocked essentially the study entity pages. So this not only gives a more comprehensive view of all the metadata associated to the study, the protocols, users can really ensure that you're aligned with verify if a study aligns with some of your research interests. And in general, if you're curious about what type of data that's being captured for all these different studies. And it's also great because there's also that loop where, like Robert mentioned, we've included DOIs across most of the studies and data sets. So this really encourages that visibility, reusability, and reproducibility for the Include Data Hub. So if you have an external article, if you click on that, you're not going to be prompted to a registration page. You're going to be prompted to come to this public study entity page. So really exposing some of that data and making it more accessible and findable. Noted that if you click anywhere else other than the studies page, it will restrict and require sign up. Sign up takes about a minute, so definitely encourage everyone to do that as well to leverage some of this data. The other piece here, as Pierre mentioned, we've added some citation guidelines that's accessible here, and we also have that information in the resource center. The other thing that hasn't necessarily been deployed, but I wanted to just show as part of a preamble to one of the talks that we're going to have later today, we've added a tile for the eMods portal, so the experimental models, specifically for mice, to drive insights in trisomy 21 and how that's being used. So that was released just recently. So you could explore that. We're going to deploy this surely today in the afternoon or tomorrow. So you could use this as a potential redirect. But Dr. Galbraith is going to be talking a little bit more into that, and he'll provide the link to the portal directly. So let's jump into it. Let's log in. And again, you could log in with either Google or OrcID. I won't spend too much time in terms of the studies page, but this is usually where you land right after you log in. So you could get a sense of all the data that's available. You could sort based on different columns here. But what I really want to highlight again is the data exploration page. This is where a lot of the effort goes into it across all the teams to really make the data across all of those studies not siloed and really harmonize it all so you can leverage these almost 16,000 participants. So you'll have some searchable fields on the left-hand side for participants, biospecimens, and data files. So you'll see here like the intervention MAXO field that was recently added, some demographics and some clinical fields. For biospecimens, you'll have things like sample type, availability. If you're looking to potentially do a request for biospecimens, we do have that option as well as part of our virtual biorepository. And also some metadata regarding the data files and what type of output was generated from the analyses that drives all of this. So we'll start off again with the use case of hypothyroidism. So you could open up this ontology browser. Essentially, you're able to search across most phenotypes based on the HP of ontology. And you'll see here that we have 1,700, almost 1,800 individuals with hypothyroidism or a phenotype that falls under the specific branch of hypothyroidism. So you could apply that, and then you'll see that it updates the results across each of the data, the table of results, whether it's participants, biospecimens, and data files. So those are all interconnected. We've added some features in terms of visualizations in the last year, which I think are really cool. So you have here essentially a type of upset plot to see essentially phenotype clusters or co-occurrence between phenotypes that based on the virtual cohort that you have built. So we're really taking the top 10 conditions here. So you can see here hypothyroidism is highly associated. Well, there's insights and patterns association with hearing impairment, which does align with some of the literature. So this is a really useful tool for some rapid insight generation, and it could pique some curiosity. So you could add that. And again, these phenotypes are added based on the direct annotations based on the participants. But once you add that in, it includes all the phenotypes under these specific, under these phenotypes. So you have the finer grain leaves under these this branch of phenotype for hypothyroidism and hearing impairment. So you could explore the results across the tables. Usually what users would do, you could explore the clinical data as Pierre highlighted under the participants tab. You could do some biospecimen requests under the biospecimens tab. For the data files tab, usually what individuals they look at, what type of analytics can I do? So here, for example, we have RNA-seq, we have some genomics with the whole genome sequencing, some BCR-seq for some of that immune response sequencing, some proteomics, metabolomics. So we really have a myriad of data modalities that are available for users, which is really powerful. And you could combine that with some features here, again, like why, for example, intervention is important. If we wanted to look at RNA-seq, for example, it is important to take a look at what type of interventions were happening. So you'll see here, like thyroid modifying hormonal agent therapy is one of the interventions that was added and applies to 60 participants. And that is something that could affect RNA expression levels. So we actually have an RNA differential gene expression analysis that was done by the Human Trisome Project team. Essentially looking at the full change between T21 and D21. So you'll see on the right-hand side, the red dots are for upregulated genes. And on the left side, you'll see downregulated genes in regards to T21. So you can click essentially the genes, and then on the right-hand side, you'll see the expression levels in FPKMs at the sample level. So that's really useful because then you could come back and take a specific gene and look at essentially, oh, I could bring that back to the data exploration page. So if I focus here on CLNBP1, that is a selenoid, a selenium binding protein that's receptor for the receptor, pardon me, for selenium. And essentially it facilitates transport of selenium, and it has been linked to thyroid dysregulation. So that's what's really interesting here is that you could select essentially the top most differentially, not differentially expressed, but higher gene expression for the specific gene. And then you could view that in the exploration page and see essentially what's going on. And you can reuse essentially some of the samples, but more so the data files if you want to reproduce something here. And this, of course, since it was an RNA-seq analysis, all of these samples are RNA, but you could essentially look at other file samples that are available. So the green check mark is something that is a biospecimen that can be requested for wet lab purposes and downstream analysis. But you can see that this specific file is a DNA sample, and there's a file, sorry, that's associated to this. So there are some genomics that are associated. So you could essentially come back here and then look at specific participants, and you'll see that there's a whole mixture of all the types of, so you have RNA-seq, whole genome sequencing, metabolomics, proteomics. So you can really do a deeper dive here. And when it comes to, so again, for the genomics, you have the VCFs, and within our variant exploration page, essentially we have extracted the VCFs of a few studies that we have. And what we do is that we enrich those VCFs, the variants that are listed in that VCF, and we enrich that with some commonly known annotations. So VAP for the most deleterious consequence, ClinVar, OMIM, NOMAD, some frequencies, some pathogenicity scores with CAD, and you could do some filters based on that. One note though is that the variant culling is based on a GATK pipeline that utilizes diploids, and so it doesn't necessarily apply for the chromosome 21. That's some stuff that Matt Galbert's team is starting to look at, how we manage variant culling for triploids, specifically for chromosome 21. But what I wanted to highlight here is that this is very quickly that this is a tool that could also contribute to some insight generation or hypothesis generation. So you can tie some of those RNA-seq data, some of the gene expression, and potentially do a combination with some of the variant data that you have here. So if you see something interesting, then you could come back and essentially do a request based on the VCFs. And you could ask questions like, oh, well, does this CELN-BP1 affect anything in terms of thyroid dysregulation in regards to hypothyroidism? Is it causative? Is it more of an immune stress response, and that's why there's higher expression? So those are the types of questions that we could ask. So very quickly, that's wanted to go over. I know I'm probably a little bit over time. So again, highlighting some of the data discovery, some of the analytics, and of course, if there's any feedback, we're definitely open to that. So please don't hesitate to ask any questions.
Great. Thank you, JP, Robert, and Pierre. We're running a little bit behind, so we're going to transition to Ariana Familiar's presentation, then we'll take questions at the end. Ariana, if you're ready.
All right. Hello, everyone. I'm Ariana. I'm a senior data scientist working with Dr. Adam Resnick at the Children's Hospital of Philadelphia. Today, I'm going to introduce kind of what's envisioned for supporting and utilizing imaging data in the INCLUDE ecosystem. I'm going to touch on some major challenges in the operations, management, and technical infrastructure as it pertains to imaging data. I'm going to talk a little bit about envision data access, usage, and INCLUDE portal developments related to supporting imaging data collections, and ending with a call for community feedback. So to just start by level setting what we mean by imaging data. So medical imaging data here is in reference to radiological data. Historically in Down syndrome research and more broadly, the major imaging modalities are MRI, CT, ultrasound, and molecular PET imaging. To date, these imaging types have been used as a tool to study structural development and differences between different populations as it pertains to, for example, anatomy or morphology. In the context of brain, for example, this could be gray matter, white matter, or structural connectivity. There's also work that looks at utilizing functional imaging measures to look at and study brain activity and connectivity. PET imaging has been used to look at metabolic changes and differences between groups. And for INCLUDE, we also want to focus on supporting the ultrasound modality, which would pertain to echocardiograph to look at cardiovascular and again, metabolic health. So for INCLUDE, why is imaging important, or what is the motivation to begin to support imaging data in the INCLUDE ecosystem? As I touched on a little bit already, imaging historically is a tool that allows us to study and quantify development and degeneration in vivo across different populations and across the lifespan. This has been particularly relevant in characterizing, for example, in Down syndrome populations, co-occurring conditions like Alzheimer's disease, congenital heart defects, or potentially immune dysregulation. For INCLUDE, the goals would be to be able to bridge the kind of patient level clinical demographic, for example, and genotypic data as is supported currently by the INCLUDE Data Hub and operations, and supplant those or be able to provide additional kind of features in the phenotypic aspects that imaging can provide. For example, you could imagine linking MRI-based biomarkers with genetic or other omic-based markers. So to date, there have been some successful data collection efforts, namely the ABCDS is a widely known existing data repository focused on primarily adults with Down syndrome. And this group or program provides access to raw, unprocessed, and processed neuroimaging data via controlled access in essentially an imaging data archive. There's also some other efforts, for example, the joint level fusion brain atlases, which are providing derivative or processed neuroimaging data, such as spatial atlases in related populations. And in some cases, these are open access as well. That said, historically, it's been relatively difficult to collect, manage, operationalize, and harmonize imaging data in Down syndrome research, but also just more broadly. The reasons for these can include the fact that imaging data is large both in data size and can be quite large in volume, and therefore the storage and computational requirements, especially when talking about longitudinal data sets where there can be several time points of imaging collected per patient or per subject. This can begin to amass quite large data sets. And this requires both technical solutions and also portal-based and more broadly platform-based integrations that can support scalable methods for data storage and release. So for example, data registration services currently are not optimized typically for large volumes of files, as is often seen in imaging data sets. And so this can create a major bottleneck in releasing imaging data. Another major challenge faced in the sharing of imaging data is proper de-identification and anonymization. So imaging and the presence of identifiable information within imaging files is pretty non-standard across data sets, even within data sets. And this is because the imaging files themselves contain complex metadata. There are many metadata fields that can contain both scientifically essential information, but could also contain, potentially contain patient level or subject level identifiable information. And so there's often a difficult balance between comprehensively removing identifiable information with over scrubbing or over removal of scientifically essential information. There can be embedded identifiable information in the images themselves that may require manual redaction, which is extremely tedious and can take a long time. And overall, these types of challenges can also cause the QC to ensure that identifiable information has been removed to be difficult and extremely manual. Another challenge, particularly across different studies, is that acquisition protocols, or the parameters for which images are collected, can differ and are also non-standard. There can be differences in image quality across different sites or different studies, even if looking at the same type of image. There can also be non-uniform file names and file structure, both within and across data sets. And so this creates extremely non-uniform and variable data organization, and it can be really difficult for investigators to utilize data, even if it is collected and shared. And so harmonization techniques are essential to capture this type of variance. And somewhat related to the data size and storage requirements are that the most scalable or appropriate methods for data transfer can require significant kind of technical personnel and infrastructure. And this is not always possible across research teams. And so in other words, the transfer softwares available to teams to be able to share their images are often non-standard, and they could rely, for example, on simple remote drives or kind of methods that are not optimized for the transfer of imaging data. Particularly, this is a bottleneck for larger data set sizes. And the last major challenge I'm going to talk about is one that is met by even the existing data repositories, and that's that data repositories are mainly focused on the storage and sharing of the imaging files. However, they're not directly connected to the types of analytic environments that researchers and data users would be utilizing to analyze, process, and extract information from the images for which they've accessed via, for example, an include type group. So this is true even, for example, the ABCDS. This group allows the data download and upload, so supports generally storage. However, there still remains to be this large burden on every research team or data user that they effectively have to download the data, store it, and handle the compute. And this creates major inefficiencies that are met by essentially every person that wants to utilize imaging data repositories. So some of the ways we are assessing and piloting being able to address some of these challenges. For de-identification, we have looked at and are beginning to establish the tools and standards that data contributors of imaging data to include can utilize to ensure the balance between removal of identifiable information and the kind of preservation of scientifically essential information in the images. And so, for example, we're utilizing a standard set forth by the DICOM-NEMA group for changes and scrubbing of file metadata. We are evaluating and providing tools for the removal of face regions of brain images as another level of de-identification. And we are further exploring the use of automated tools, for example, AI-driven tools for image redaction of pixel level PII, PHI, and additionally text-based detection of potentially any remaining PHI, PAI after de-identification pipelines and processes have been run. We additionally are establishing what standards would look like for the release of imaging data sets via INCLUDE. Depending on different levels of de-identification, for example, there may be different levels of access control, and typically what would the appropriate mechanism for access will be going through dbGaP for controlled access, and potentially having an additional layer of registered tier access, depending on the data set in hand. For harmonization, we have assessed and provided imaging QC tools and reports to be able to at least begin to review the kind of cleaning and harmonization that happens upon data ingest, as well as the de-identification level. We are further developing out automated tools for image validation and harmonization techniques. For example, there is an image classification method that can be used to extract meaningful and harmonized labels associated with each image, defining what the image type is. And we have methods of then feeding those into, for example, the INCLUDE portal or other methods such that those labels can be used for exploration and use of the data downstream by data users. Importantly, this generalizes across the variants in acquisition protocols and naming conventions, as I mentioned earlier. And ultimately, the goal here is to both support the ingest and the support of data contributors, as well as the rapid and secure release of imaging data and integration with other data types through the INCLUDE portal, and the use of that data downstream. This we are and have been exploring cloud-hosted storage file and establishing metadata standards such that the kind of storage and compute environments are user-friendly and efficient. And as I'll speak to in a moment, we have piloted and are kind of establishing what the interoperable platforms look like for downstream analytics to, again, fully enable the user to not only just access and download data, but also utilize the kind of specialized modality-specific tools that are essential for data processing and analysis. Okay, so in terms of portal developments, the goal is to incorporate imaging with the other existing data types in the INCLUDE portal. And this would allow users to search through the imaging data collections available that have been made available and for which they can request or gain access to. This also allows users to create cohorts based on combinations of imaging as well as other modality features and facet types. And additionally, what we aim to include are some of those harmonized classifications for the different imaging sequences, different imaging scan types, to again, just make the use of the data, especially at larger and larger scales, more user-friendly. The goal then would be to allow users to select, of course, gain access to, and then port the files of interest for their given study into analytical environments that can support data modality-specific data visualization, and kind of pipelines and processes and third-party software tools as desired by the user. And then additionally pull those derived features and files into uniform downstream environments where users can use their kind of favorite data science tools of choice. And so again, this really is to support and leverage existing infrastructure and ecosystem tools to allow users to kind of focus on their scientific exploration of interest and really alleviate the technical burdens, as I mentioned earlier, in terms of data storage and compute. Again, this is to really just bridge the ability for users to both access and analyze imaging and multimodal data across the suite of existing platforms that enable these integrated analytics. And with that, I'll just end on the request for feedback from the community. So if any of these kind of speak to you, please don't hesitate to contact the team via the form or the email provided at the bottom of the slide. We would like to request feedback on whether teams or individuals have existing imaging data or potentially collecting imaging data that they see as valuable for contribution to the INCLUDE repository. Are there research studies or use cases that you might benefit from with access to imaging data? And additionally, if you are using or plan to use imaging data in your research, are there specific needs or requirements in terms of data management, analytics, or tool development that could better support you in your research? And with that, I'm not sure if we have time for some questions or we'll move on to the next section.
Thank you, Ariana. We're running a little bit late, but let me pause here. We're halfway through, only 3 minutes exceeded. See if there are any questions. I have a smaller group today, so if anybody wants to unmute or pose your question in the chat. So Jennifer in the chat is asking for the URL, Ariana, for the feedback.
Okay, yes, I'll add that.
Very good. So if there are no questions or comments, we'll keep going here with an exciting presentation from Matthew Galbraith about the experimental models portal.
Okay, good morning or afternoon, everyone, depending on where you are. Yeah, I'm pretty happy to announce today the launch of our experimental models portal of Down syndrome. I will share the link afterwards, so you don't need to worry about copying this down right now. Really what this is designed around is an area that the INCLUDE DCC hasn't focused on, to date, which is experimental models of Down syndrome, right? The vast majority of everything that's in there at the moment is centered around human subjects data. And so we are aiming to serve that part of our research field. And so again, just by way of introduction, I shouldn't have to explain this to many people on the call, but so we're all on the same page. We know that trisomy 21 affects essentially every organ system and tissue in the body, right? And this is just a quick survey, certainly not complete. But the easiest samples for us to get hold of, and many of the samples that are currently in or data generated that's in the INCLUDE Data Hub, is based on samples obtained from blood or plasma derived from blood. And while they can tell us a lot of things, in particular, the plasma, of course, tells us about all sorts of different organs that can secrete molecules into the blood, proteins, metabolites. We can study the immune system very well from the blood. How do we expand upon that? Given that we know that trisomy 21 affects nearly every tissue, it's fairly difficult to get some of those samples, and in many cases unethical. Moreover, we really cannot adequately establish cause and effect there. And so that's where we need to turn to experimental models, right? So we can study these complex cause-effect relationships Of course, we can use animal models of various different flavors, and other avenues like iPSCs, induced pluripotent stem cells, or other cell lines, right? And so clearly data from these models is really important for investigating mechanisms for testing preclinical applications of things, right? And so in my research group, we're really interested in, these impacts of trisomy 21 across different tissues, across development, perhaps even across different models. And I'll cover that in a little more detail here shortly, what I mean by that. I'm just going to attempt to change windows here, and this is actually the live portal. And so on our landing page here, our home page, we have these different tiles, and these are really designed around different user journeys to try and fulfill different needs as we surveyed the research community, particularly people focused on experimental models. These are by no means the only user journeys or entry points that we're going to remain on the site. We're certainly working on others. I'll mention that a little bit at the end. But really what we have are sort of two areas here aimed around sort of having a knowledge base here to find publications. I'll demonstrate that here in a minute. There are two different entry points into some of the omics data sets. We also have a section providing information about the animal models, which can be quite complex to understand, including phenotypes measured in different animal models. This one is coming soon. We're going to have information about cell line panels that have been made available by different investigators that you can request here from the site. And likewise, we have a collection of mouse biospecimens from the team at the Linda Cernick Institute for Down Syndrome, where you can go in there and explore those and request samples from that collection. And we're hoping to get other collections here, either under the cell lines or the mouse biospecimens. If you have a collection you'd be willing to share, please do get in touch. We'd love to talk with you and discuss how we can make that available to the wider research community. As always with Include DCC, we're open to feedback. We encourage it. There's the same button here that you'll see on the main Include DCC data hub that you can take a screenshot, give us feedback that will come straight to us, and we will try to address those questions, concerns, comments, feedback. Great. So I will dive here into our study finder, and the vision here was to help you guys find publications that are relevant to experimental models. Right, if you go to PubMed, it can be quite difficult to find all of the publications around a particular mouse model. There's myriad different ways of specifying the name of that model. There are colloquial terms, and then there's the full genetic description. And so you can do a free text search here. We can search, for example, for the DP16 mouse, and that will then live filter to the publications. You can see this is chosen filtered to 45 here, but you don't always get the same answer. And so that's our quick way to do this. But what we have done is we have curated this set of publications, and we've again focused initially on Include funded studies and other important studies in the field. We may not have caught everything. You can tell us about more by clicking this button. I'll show that in a minute. What we've done is we've really curated these publications so that you can select all the publications that say use a mouse model or human iPSCs. And so if we chose the mouse model, you might be interested only in publications with the DP16 model, right? So now you know every possible version of typing or spelling out or specifying that model, you've now captured that here. And likewise in PubMed, it's actually sometimes not even that easy to choose a specific assay or data type. I'm going to choose RNA sequencing here, right? But you could write that out as transcriptomics or single cell RNA sequencing, so on. So again, now this has filtered us to the publications that we have annotated with that mouse model and with bulk RNA sequencing. So you know that you've got those. You can click through into these, and then that gives you some information about the publication. You can link out to PubMed or the digital object identifiers. We will have in the near future the ability to link these through to any data sets that are in this portal associated with that publication. So I'm going to go back now to our homepage. So we hope that that's a useful tool. Again, you can hit the feedback button there. Actually, I forgot to demonstrate one other tool here. If we've missed a study that you know about and you would like it to be included here, you can hit this button and tell us all about it. We'll get that into the portal in one of our future updates. Please do provide the PubMed ID. That's super important. You can help us with some of the tagging. These are really asking the questions about the tags we use, and that will help us to keep this current and expand things to make sure we're capturing everything that's out there. which is somewhat of a challenge. So now I'm going to dive in, and what we have here now are some tools to visualize data sets generated from experimental models of Down syndrome. And so perhaps you have a favorite gene, protein, or molecule, and this is the entry point you would choose. And so you might be interested in interferon receptor genes. This is FNAR1, so we can select that. You can choose mouse or human genes. So that's going to load up quite a lot of data here. What it's going to tell you first is the number of data sets from each tissue where this gene has been detected. So you can see there's a number here. You can now come down here and select those data sets and get a live view. So I might be interested in expression of this gene in the whole brain of the mouse, right? And so now you can see from this particular study, was that significantly upregulated or downregulated? In this case, I'm cheating a little bit because this is a triplicated gene, but clearly that is upregulated or more highly expressed in the DP16 animals compared to their wild type littermates. You can compare across studies. So maybe you can see here there's a different study that also has measured that gene in the brain. And so we can see if these two studies agree on what's going on here. And in fact, in this case, they do. Perhaps you want to see different brain regions. So we can do that as well. And so you can select up to 6 different rows from the data table here and compare those results. We're not reanalyzing the data here currently. We may do so in the future, but this is just reporting the statistical results from those 3 different publications. So you can see and explore what your favorite gene might be doing across different mouse models. We can also perhaps you're interested more in developmental time points. So you can see here we have 3 different heart tissues at different time points, adult and 2 embryonic time points. We can compare that. And you can explore whether your favorite gene might be upregulated across developmental time or perhaps only at specific ages of the mice. Right. So perhaps you don't have a favorite gene. Maybe you have a favorite tissue that you're interested in. And so this is the same underlying data, but you can take this entry point now. And again, to keep things easy, I'm going to go just with the whole brain here. So now you can look up and focus on all of the data sets that are available. In this case, bulk RNA-seq we've chosen. We can select up to 2 data sets here to compare. And what this is going to do once the data loads is going to show us the most significantly dysregulated genes in those 2 data sets, right? So we can see here, and what we have done here is we've colored the triplicated genes, and you can see those are almost exclusively on the right of this plot with the x-axis being representing fold change. Here's this FNAR1 gene we were looking at just before, but perhaps you didn't know that and you wanted just to see the most upregulated genes. You can click on that dot, and now that will show you again the same underlying data as the other view, what is happening there. So this we're hoping helps people explore, maybe even get as far as finding some preliminary data about their gene or tissue of interest that can help them start on the journey of selecting the appropriate mouse model or even preliminary data for grant applications, so on and so forth. So we'll go back out to our homepage again now. And our next section is sort of part of what we consider the knowledge base portion of the site. We'll go in here and learn about the animal models. And so in this case, this is focused for now around the mouse models, but we have future plans to introduce information about the rat and any other models that may be relevant here. You can search for a gene. And so for those of you who don't know, the mouse genes that correspond to human chromosome 21 genes are actually found on 3 different chromosomes in the mice. This makes it rather complicated to model and has led to a proliferation of different mouse models of Down syndrome that triplicate varying numbers of genes that are orthologs of human chromosome 21 genes. In some cases, they triplicate additional genes that aren't related to the human chromosome 21 genes. And so it can get quite complex to navigate this. And so you can choose your favorite gene here, and it will highlight for you which chromosome that gene is encoded on in the mouse, and therefore give you some information about to guide you in selecting appropriate mouse models, right? If you're interested in APP, you can see that would be triplicated in the DP16 mouse model, and then these 2 here, TS65DN and TS66. And so that gives you, helps you narrow down. You can here, again, link out to the original publications on each of those mouse models and link out to the JAX lab where you could order those mouse models. You can download a list of the genes that are triplicated in those mouse models. The other piece that some of our users that we interviewed really struggled with in getting into this field was understanding what's been already reported or in terms of phenotypes in each mouse model, or how to relate that to human phenotypes. And this is perhaps one of the more ambitious aspects of the entire site, because there's a lot of different ways of reporting this. But what we've done here is attempt to capture from, again, a curated set of publications, the phenotypes reported in these various mouse models of Down syndrome. And then we have mapped those phenotype observations to an ontology here. In this case, it's the mammalian phenotype ontology. And really, this is just sorted by the number of observations in our database. And so we can go here and drill down in this tree. If it's green, it means there's a direct observation at that level. Otherwise, you can drill down into those different nodes. And so we might choose something like abnormal learning memory conditioning. And you can see here, you can link out to more details about that term. You can link out to the equivalent human phenotype ontology term, HPO. This is actually what is used in the main Include DCC hub to classify the co-occurring conditions and other phenotypes observed in the human studies. So this immediately helps you to relate those together. We're going to provide more direct links in the future. And you can see in this case, this phenotype has been reported to be present in the DP16 animals. You can click on here and get a list of publications from which we derived those observations. And you can see that one was supported by multiple publications. That's not the case for everything. We can see that it was reported as not being present in the DP10 and DP17 models. And it was reported in this TS66YAR, and then in fact this TCMAC21, which is a humanized version of a mouse model of Down syndrome. So what we're hoping here is that helps people to understand some of these various phenotypes and how consistent they are across different mouse models. Now, comparing DP16 to 10 and 17 is a little bit unfair in this case because they don't triplicate the same sets of genes. But here, comparing DP16 and TS65DN and the TCMAC21, we know that they triplicate many of the same genes. And so you can see whether that sort of corresponds to something that is a consistent phenotype versus something that perhaps hasn't been assessed or that we haven't at least been able to find reliable reports of. So we hope that this will be a very valuable tool for the community. This is the one that's a big challenge to keep current, as well as making sure that we've captured everything in the field. So if you know of something that we've missed, please do hit this button here. And just like on the study finder, you can tell us about those phenotypes. Very, very difficult to capture the entire state of a field here. So please do participate there if you have an interest in that. Keeping an eye on the time here, I think I can take us through pretty quickly the last section of the site. So again, you can enter that from the menus up the top here, or from these tiles on the home page. And this is our virtual biorepository. So this is where we have a collection of mouse biospecimens hosted by the Linda Cernick Institute for Down Syndrome. We're hoping to have more collections in the future. If you have a collection you'd be willing to provide, please do get in touch. And you can see here that we have a number of different mouse biospecimens that are available for request. Of course, that does not guarantee you access to those samples. We will get some information from you and judge whether we think that that's a great idea. These are very valuable samples, of course, but we're really willing to make them available to the community, help people get started. You may not need to start up your own mouse colony, which has considerable time and monetary costs, right? So you can come in here, you can filter for different animal characteristics. You might choose a particular mouse strain. Maybe you want a particular age group. You want to make sure we have various treatments, interventions. I'm going to choose none here, and that's going to load up. So we can see that we still have 477 biospecimens available from the DP16 mice that haven't had any sort of treatment. And then you can choose sample characteristics. So we might want a particular tissue here. Let's say we'll stick with our whole brain. And that's now going to filter down. Now there's 42 whole brain biospecimens in the freezer. And then you can select whether they were fixed or frozen. Let's say we want flash frozen. And you can see what's available there. And you might want to select some of these rows. You can click on some of these rows. You don't have to. You can also make a request for everything that's eligible. And then you hit this button here, and that's going to ask you some questions about who you are and what you might want to do with these samples. And so that will help us sort of judge whether we agree that this is a great idea and that's a good use of the samples. And then that will send a list of the samples to yourself as well as to our team, and then we will be in touch to discuss that request with you. So again, we really hope that this website, this new portal, helps to serve the experimental models focused part of our community. We are going to have list of these cell line models available, particularly the iPSCs. Again, if you have a collection of those that you'd be willing to make available to people, we can help you publicize that. So I will end there. Happy to take questions either now or at the end of the session.
Thank you, Matt. Checking the chat here. There any questions?
And I've just dropped the link in the chat there too, if you want to check the site out.
Very good. Yeah, we do have some time for questions if there are any before we wrap up the webinar today with a presentation from Dr. Costello. I don't see any hands up or any action in the chat here. So I guess that's our signal, Jim, to yeah, proceed with your scientific presentation today. Thank you for joining us.
Yeah, sounds good. So hopefully you can see the correct presentation. Yeah, so thanks for inviting me to give a talk today. And what I want to cover today are two separate topics. So I'm going to hopefully spend about 10 minutes on each of these topics. Both of them are oriented around trying to understand, at least in the context of multiomics data, how do we integrate that? How do we bring more pathway information or process information into that analysis directly? And then the other one is a large language model that we've been working on in the lab that's going to be basically trained and very specific to Down syndrome research or include funded projects through that. So I'll start with the first methodology. And whenever we get these large multiomics data sets, we get a whole bunch of gene expression by a whole bunch of different samples. Oftentimes, one of the first types of analysis that we'll do is we'll try to take all of that information and project it down to a space that we can actually look at and understand. So analysis like principal component analysis, PCA, this is a very common methodology that we use. PCA, basically, again, it takes that multi-dimensional data, it puts it in two-dimensional, three-dimensional space, depending on how you want to plot it. And then those individual points represent, essentially, in this particular example I'm looking at, it's going to represent different cell types, but those could represent different individuals. It could represent, different samples that you're trying to look at. And each of those components represent the variation in the data that we're trying to look at. So oftentimes what we do is we'll do these decompositions. So principal component analysis or a UMAP is another very common one that we use, especially with single cell data. Another method is called non-negative matrix factorization. Again, all of these methods are attempting to take this big kind of multidimensional data, project it down, and then oftentimes what we'll do is we'll try to project on information onto those components. So in this case, again, we're looking at different cell types, so T cells, NK cells, B cells, myeloid populations. And you can see that those principal components or the variation we're looking at the data, we can map the different cell types on there, and then it makes a lot of sense. But oftentimes, these principal components or any of these components we're looking at, they don't necessarily map one-to-one with the biological interpretation. They often will be fractions of, it might partially explain, the cell type or maybe the treatment and maybe some other biological factors that are in the data. So keep in mind that this is just a mathematical decomposition. Ultimately, we want to get to the point where we're looking at, for example, this is looking at a breast cancer data set where in breast cancer, we can break apart the individuals into different subgroups or subtypes that allow us to then think about different therapeutic options for those individuals. So, and oftentimes we can break those apart by the gene expression profiles, and we can also think about how the different underlying, so for example, the basal lineage or the HER2 lineage, there's different molecular pathways or processes that are important or driven or help drive those different subtypes. So ultimately, what we would really like to be able to do is take that big expression data matrix, decompose it into those different subgroups, and understand what are the molecular pathways or processes underlying or driving those individual components. So Lucas Gillenwater, a graduate student in my lab who's recently graduated, came up with a methodology where we were trying to incorporate both sample annotation, so that metadata information that was previously being discussed, and also gene regulatory network information into that decomposition. So the whole idea is that if we can bring this information directly into the calculation, then once we get those subgroups that come out of there, they're going to be better aligned with the biology we want it to be aligned with. So in a sense, we're trying to bias the decomposition so that we get out more interpretable or more easy to interpret types of results. This is the math behind it. This is the only equation I'm gonna present today, so don't worry. But in short, what we're trying to do under standard decomposition is this Y matrix, so your input matrix here, is being decomposed into this W and this H matrix. And ultimately, we're trying to minimize the error in that decomposition. So that's basically what this is what this says here. And our adjustment is basically we're taking into account this sample information or this gene regulatory network information, and we're weighting that bias depending on these little lambda values, lambda 1 and lambda 2. So we can weight more for sample information, we can weight more for gene regulatory network information, and how that is affecting the decomposition. So how do we test this? How do we know if the methodology is actually working? One way to do this is through simulations. So we can kind of generate data based on prior knowledge, and then we can see how well the methodology is able to reconstruct that data. So we take a biologically informed network. So this is a network that we know how the relationships of the genes are related to each other. Then we can look at little kind of subgroups within the network, and we can manipulate those computationally to overexpress certain genes or groups of genes in the case of Down syndrome, and see how those little groups of genes that we know are important and regulated together affect the output or the gene expression profiles. So that's exactly what we did. So we took this regulatory network, we identified certain modules within the network, and then we did a simulation, and we essentially overexpressed or underexpressed or manipulated the expression of the genes within these different subgroups to create our training data set. So again, we have gene expression profile, little subgroups are being manipulated, we know what group those belong to, and essentially we can train our model on this data and evaluate on a testing data set. So the testing data set was generated the same exact way, it was just held out so that we wouldn't bias the model. So we take the sample information, we take the gene expression matrix, and then we can factor that into the decomposition, and we can evaluate how well we're doing. So what does that look like? So if we were to simulate a whole bunch of gene expression data using this particular approach, this is an example. Again, this is a gene expression profile. This is hierarchically clustered. And what you can clearly see is that there's little patterns that pop out of this that are associated with those different sub-networks that we mentioned, that I talked about in the previous slide. So very clearly, if I have a very clear signal in the data, hierarchical clustering can really nicely group those individual subcomponents, and we can see that the subtypes break out really nicely according to these colors. But as we add in more noise or more disruptions into the system, we can see that those patterns basically completely dissolve. And when we try to do hierarchical clustering, we completely lose that subgroup structure. And this is probably closer to what we see in the real world versus very, very clear examples that we see over on that kind of left side of the plot. So how do we evaluate this using real data? So what we did was essentially we used gene expression data from breast cancer, because we know the answer quite well. I'm sorry, in this case, this is all simulation data. We get into breast cancer in the next slide. And we look at how well we're able to identify those subgroups that we defined previously. And we can factor, and we can weight again based on sample information, based on gene network information, and then we can evaluate that across multiple modalities. The short of the story is, even as you add in more noise into the system, this little kind of, what color is that? The little kind of orangey reddish color here, that's the Grackle method. The green would be if we only regularized on gene networks, and the blue would be only if we regularized on sample information, and this NMF is just your standard no factorization at all. So in what you can see is we want a higher performance. So we see that even as we add in noise, we're still able to have better performance, either if it's the average of the performance across all of these values here, or if it's the best individual model, we're still able to replicate that input data that we talked about. So again, we moved to gene expression within the breast cancer space because we wanted to see how this would work in kind of like a real world data set. We evaluated if the model could basically replicate or identify those subgroups, those subtypes in breast cancer using previous annotation. So this is kind of a sanity check. Can we identify the subtypes given subtype information? This is looking at, can we identify the subtypes using methylation data, so a different form of data to identify the groups. And this is kind of a multi-omic kind of integration approach that we use. And in all the cases, again, the Grackle model is able to better identify those subgroups compared to the other models or just a random version of this model throwing relationships together. So we mentioned, so that's all sample information. So the other side of that is the gene expression information or the gene network information. So if we were to look at different ways of constructing networks, there's lots of information out there. So we did a kind of an inference method called Panda. There's also a pre-annotated database. And we could see that the performances, are pretty close, but the performance of the model is also dependent on the underlying network structure, which is kind of what you'd expect. So this is obviously that model was placed, and I showed you breast cancer data simply to show you kind of a very well-established relationships that we already know about, and can we replicate those? We applied this to HTP data, so the Human Trisome Project data, and looked to see if we could align those different kind of groupings that come out of there, so the individuals that are grouped together. And then we look at, okay, so this methodology picked out this group of individuals to be grouped together. What do they have in common, and how are they different from other groups that were identified? And in this case, this group was identified as having over-enriched for hypothyroidism and obesity. And then what we can also do is we can look at the underlying genes and how weighted they are in what we call a latent variable, okay? So again, the point here, I'm trying to explain this in very kind of abstract terms, but we're trying to identify groups of individuals and understand the underlying genes that are driving that grouping based off of that regulatory network. So we can kind of rank order those genes, and we can see something like KIA 00040, which is a gene that probably most of us wouldn't even think about. But this has been previously shown to be involved in glioma growth, controlling for JAK-STAT kind of signaling, which would be JAK-STAT signaling within Down syndrome is highly relevant. So it's a discovery tool. It's trying to identify groups. It's trying to align and kind of rank or prioritize genes that might be very important in those conditions. And then we can go in the lab and actually follow up and study those. So again, I have a very little amount of time, so this is just one example that I'm talking about. I'm happy to answer any other questions after that. The second part that I want to talk about, the second methodology that we're kind of working through, is building a large language model specific to Down syndrome. So hopefully everybody is familiar with at least one of these platforms. So ChatGPT, Copilot, Grok, the LLM model for basically Meta or Facebook. Claude is another really popular one these days. And, essentially, these models have been amazing at being able to generate text. So they're generative models in answering questions, right? And they've been applied, and they're starting to be applied more and more to the biomedical research side of things. So, for example, we can use ChatGPT-based embeddings to look at kind of single-cell biology and to analyze single-cell RNA sequencing data. This has been used in kind of the CRISPR space to identify guide RNAs that would be highly relevant to any kind of target there. So, really what we want to be able to do is to take those models, take the power of those models, and be able to use those in the context of the 48,000 plus publications that are specifically looking at processes in Down syndrome, basic research, experimental models, clinical trials, all of these things are on target. But I do want to talk about why we need to do this specific to Down syndrome versus just using ChatGPT or just throwing ChatGPT on top of the data portal itself. So, in a generative model, these models are essentially looking to predict the next most likely word, or most likely word relevant to some conditions that you give it. So for example, this is, an example from a previous publication, but it says, tell me something about llamas. Llamas are, what's the next word? The next word in this kind of string of words is very probably domesticated. So then it's going to put domesticated, and then it's going to continue on adding those words as it kind of builds the response to the query. So oftentimes these are called hallucinations within kind of the LLM space. These aren't necessarily hallucinations. They're what the model is trained and predicted to do. So, there's no mystery here. This is all basically probabilities of likely words to come based off of how it was trained and the data that it was used to train. So if you have outdated training data, it might give you bad information or false information if the information it was trained on isn't up to date with kind of the current research. It can lack domain expertise. So we could talk about generalizations because, most of these LLMs are trained on things like Reddit and Wikipedia and other websites that have information that it's able to pull in and kind of build its queries from. So there's no, if you're training it on all of Reddit or Wikipedia, there's nothing specific to Down syndrome, and it might lose the context of looking at Down syndrome versus just training on everything. Poor attention to mechanisms. So these models are trained on text, oftentimes comments and thoughts. They're not thinking about very detailed molecular mechanisms or how things are related to the biology. And then it could be, and then the prompting is also really important. So if your user queries, lack specificity, or they're giving you very general kind of questions, like tell me about Down syndrome, like that is just, it can go in any direction from there, and it doesn't really know where to go. So an example would be like, the XS gene on chromosome 21 is responsible for Down syndrome. Well, that's factually incorrect because we know the XS is in fact on the X chromosome, not on chromosome 21. So this would be an example that is structurally correct. It looks like a reasonable statement, but is actually a false statement based on the actual biology. So how do we approach this? So if you look at a typical large language model, what you do as a user is you write your prompt, you write your query, and then it goes and hits that pre-trained large language model. And that was, again, trained on all of these previous, anything on the web, there was a recent issue with Anthropic, which is the Claude model, where they downloaded essentially a huge library of books and trained it on that. So it depends on the corpus of data that you're actually training it on. But you hit that pre-trained model, and then it's going to give you a response back based on all of the knowledge that it kind of gained. There's been updates more recently in thinking about how do we actually make these systems a little more focused or targeted towards the real query you're after. And these are called these RAG models, so these retrieval augmented models themselves. And essentially the adjustment here is that we take a query, we have a retrieval model or a retrieval system, which hits a knowledge base, a specific knowledge base, and returns back the relevant documents to your query. And then we leverage this really powerful large language model and being able to say, okay, here's my knowledge space, here's my query, put those two together using all of this great ability to generate text and give me back a response. So this is what these RAG models are really doing. And we can have multiple agents, and we can really train these models to be very specific. So it's a little complicated. I'm just trying to give you the big picture idea here. But the process that we're going through currently is taking Down syndrome related peer-reviewed papers. This work is being led by my postdoc, Sutanu Nandi, whose picture's on the next slide. We're going to specific publications that we know are funded through INCLUDE or that are highly relevant to the research we're interested in. We're taking all of those papers we're breaking them apart by like abstract results, methods, et cetera, et cetera. And we're embedding them. So we're basically making them fit into some of these large language models so that they can be queried over. So we embed those into the model format, and then we store them in a large database, okay? And again, this is the database that we're going to query against when we actually run our query. So we fetch the PubMed central IDs. We basically take that XML formatted data, transform it. We split it apart. We use, in this case, we're using the OpenAI. So this is the ChatGPT platform, using the text embeddings to create kind of the vectors so that when we use ChatGPT, we can kind of match and query over that. And then we're going to store those embeddings in this database. Currently, we just started the process. We have about 420 papers that are present within the database right now, but those are chunked up into about 13,000 different chunks. So then ultimately the system looks like this. We take our query, we basically take that query, we embed the query so that it can be used and queried against the database. We find papers, processes, or references that are very similar to the query that match a certain criteria. And then we take those number of papers. So we take the query plus the context, so the query plus the papers, and then we give it to this large language model to then generate the response back to us, okay? And we can structure the internal kind of workings of this model to say, I want a summarization agent. So I want something to write a summary for me. I want something that's going to basically take the citations and format those for me. I'm going to re-rank. I'm going to basically take the paper and say, how relevant is this? And I'm going to give you the score related to how relevant it is to the query. And then we can format that output however we want to. Okay, so then this is just an example, this is a real example. This is actually queries done. So what we're going to do is we're going to take this query, tell me how STAT1 is related or potentially causal in any conditions or diseases expressed in individuals with Down syndrome. So, and I use this specifically because we know that STAT1 is an interferon response, right? Both it's responsive, responsive to interferon signaling, regulates a whole bunch of downstream interferon response related genes. And we know that those genes are involved in conditions related to Down syndrome. So this is we already know kind of the answer to this. So we want to be able to check to see how do these models respond to those queries. We're going to look at 3 different versions. So we'll look at ChatGPT first. So if we look at the response from ChatGPT, ChatGPT says, okay, here's a summary on how hypothetical STAT1 is possibly connected. It talks about the background of STAT1, interferons, and immune signaling. It's like, yeah, this is good information. Yes, STAT1 is related to interferon signaling, JAK-STAT signaling. STAT1 is related to kind of immunodeficiencies, autoimmune-related conditions, et cetera, et cetera. But ultimately, this isn't very specific to its function in Down syndrome. It's providing general information, which is exactly what you'd expect from a very, very general model, right? It also doesn't provide you with the actual scientific evidence. It doesn't provide you with citations. It doesn't point you to where you found this evidence from. It's just summarizing kind of the gestalt of what's out there. So you can't go and verify what some of these claims are. Claude is another one, same thing. We throw the query in there and see what it says. This one's maybe a little bit better. It talks about Down syndrome is, a condition that with the trisomy of chromosome 21 contains genes that are interferon responsive. So, it's starting to make some of these connections, but what it says in the end is that, in summary, while there is no direct causal link established between STAT1 and Down syndrome, the involvement of STAT1 and blah, 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 blah. So it does make some kind of inferences from that. Again, no citations, there's making speculation and can't verify the resources. So this is our model. So we're calling it the T21 Research Assistant. So again, this is for research purposes at this point. We're trying to summarize information, potentially generate hypotheses, provide key evidence. So when we ask that same question to the trisomy T21 Research Assistant, what we see is a summary that is much more specific to Down syndrome and how those STAT1 is related to JAK-STAT signaling within Down syndrome. And we provide the actual direct references associated with each of those statements. So if we can make a statement about JAK-STAT signaling pathways, which are associated with high rates of autoimmunity, it's very blah, blah, blah. We can point to the publication and the area within the publication that it was actually referenced. So, and you can see how this kind of breaks down. We have key evidence, we can generate hypotheses, we can provide additional summaries, we could provide additional kind of information on top of that. And again, right now we just have only trained on a limited set of data at this point. Okay, so that's really what I wanted to talk about and show you. Again, I'm already over 20 minutes just with that short presentation. Just a quick thank you, Costello Lab, Green Lab, Pivadori Lab. So we're a group that was funded through the Include to kind of build some of these tools, and that's what we're doing. So we'd love to work with the Data Hub. We'd love to work with EMONS to get some of these tools kind of built into those systems. There was a lot of references to being able how to use these systems, how to find information, and hopefully these tools can be used in that way. And I'll stop there.
Thank you very much, Jim. Great stuff. Very good. We do have time on the clock to entertain any questions or comments about Jim's presentation or really any of the presentations today. I'll be monitoring, hit the chat, but again, be mindful that it's not an exorbitant amount of people today on the call. Feel free to unmute yourself if you have a question or a comment. So Jim, I have a question. What the assistant that you guys are developing, providing all the right citations. If you go to one of these other models, and in your prompt you ask, and give me the citations. Right? If you create the prompt in a way that you ask ChatGPT, tell me about a STAT1 relationship to Down syndrome, please back up any one of your claims with a citation. Is it capable to do so?
That's a good question. I can run that experiment in real time. So give me 1 second and I will tell you what it does.
Very good.
In fact, I'm literally doing that right now. Let's see.
Hi, this is Catherine Koenig from the Cleveland Clinic. I just wanted to say that I've tried this too with ChatGPT, and I find that it makes up citations that don't exist.
They
So
look real. They look real though.
They look very real, yeah, but they're not real.
Yes. So that's what I was hoping. Oops, so it looks like it's actually searching some of these. The newer this field is moving so fast as well that what I say yesterday might be different today. So let's see what it does.
Yeah, while we wait for that too, one of the things that we're trying to do with some of these tools, working with Jim's team, is that little innovation that he just showed you, which I think you made a little bit of light of there, Jim, but it tells you which section of the manuscripts it comes from, right? Knowing whether it comes from the results or the introduction is actually quite important. If you want to summarize new information, it needs to come from the results, right? From the introduction, they could be summarizing 100 other papers. And so knowing and being able to report back where something came from is quite important. And something else that we're working on is perhaps we want a version of this that really focuses on just the experimental models, right? And being able to have those other layers and tell it to prioritize things from experimental models could be very useful, right, in that context. And this is something that we're hoping to incorporate into the experimental models portal in the future.
Yeah, well, here you go. ChatGPT is getting better. So they at least found Matt's paper, they found Kelly's paper, and they do reference those. So it does provide, and it does provide a link out. So, even from when we started developing the system, they've already gotten better at it. Yeah.
It's still a very generalist thing though, right? It doesn't sort of provide as much context as what we're hoping to be able to do with these sort of tuned versions of this.
Yeah, and we can link it directly back to EMOD. So, if you wanted to say, okay, well, what, it's coming from this paper and there's this result. Now the gene expression, we can link back and provide some additional kind of context and analysis as well. So I think there's still value in it. ChatGPT hasn't solved everything.
Very good. Any other questions or comments? We don't have to use the full hour. We have another 10 minutes or so. We can always get some time back. Anything else for the group today, including the earlier presentations? Well, if not, I want to thank all the speakers who presented today. Thank you, Jim, for coming and visiting today and presenting that is interesting work. And yeah, please do subscribe to our newsletter. Stay attuned for various events and developments. And if we don't see you before then, have a great holiday season. We'll have a spring webinar for sure. Thank you, everybody.